welcome everyone to this BSMT conference, a live webinar. I'm very delighted to welcome you to this event. First of all, I've been given some strict instructions on what I'm to do, and we'd like to thank all our sponsors who are on your screen now, but with their support that's made this event possible. I'd also like to thank our two speakers, Catherine Moore, who you can see at the moment on your screen, and Alan McNally for doing this talk, preparing all of this and giving us some really fascinating insights into COVID diagnostics. I'd also like to thank the BSMT committee for all their work of pulling together a fascinating programme and giving the information out in this sort of challenging world and getting us all together, even if we're not all quite in the same place that we normally meet together. The webinar is going to be recorded and will be available via electronic links later on today. There will be, if we don't get round to responding to all the questions will be responded to as well by the end of today. And I'd finally like to very much thank Professor Eric Bolton, who was the previous president of the BSMT and has just stood down, and somehow at a weak moment I volunteered to be the future president of the BSMT. And I look forward to taking on this role. So at this point, I think that's very much enough from me, and I'll hand over to Catherine to talk about what she's been up to for the last year or so.
Thank you very much for inviting me to speak about how we set up and run the COVID-19 testing service in Wales. As you can see, it's not just my name on this starting slide. I've also got Rachel Jones, who is our clinical lead in Cardiff and also the Welsh virology lead, and Jonathan Evans, who's the technical head of the Wales Specialist Virology Centre. They also contributed to this presentation. So my opening slide is really to show what we looked like pre-pandemic. So I work for Public Health Wales. Wales has devolved, was one of the devolved administrations. We are devolved in terms of health and education, for example. Public Health Wales is the national public health agency for the population of Wales, so we look after the health boards. So we provide direct microbiology services for 5 out of 7 of those health boards through a network of 8 laboratories. The virology advice and reference services are centred around the Wales Specialist Virology Centre in Cardiff, but we do get support from the Swansea laboratory in that work. All microbiology has standard operating procedures, so that includes any laboratories which are not within our Public Health Wales Microbiology Division. So those are the labs in Newport, in the Royal Glamorgan, which is just north of Cardiff, and down in Withybush, which is right down in the southwest of Wales. And we all work on a single laboratory information system, which means we're entirely connected across Wales, and means that, so for example, we can in South Wales authorise results generated by laboratories in the north. So when the pandemic arrived, we had to upscale our testing quite dramatically, and this involved upscaling the service in general. So we went from our 8 laboratories, which we implemented with medium and high throughput testing, including some of the screening service, to include 7 regional hot labs for rapid testing. So these were set up in DGHs and hospitals where they had no microbiology laboratory, but we sort of implemented a single room with lots of different sort of black box technologies to allow them to test locally. And with the non-PHW labs, we also equipped those with medium high throughput testing, and we also have one of the DHSC Lighthouse laboratories in Newport, which I will come to later. So all of these Welsh NHS labs, so including those outside the network, follow nationally agreed SOPs, and all of those results that we have feed into the national LIMS system to enable us to see results from across Wales. The Lighthouse Lab data from the UK, if it has a Welsh postcode, also comes into Wales and feeds into that common centre so that we can see results from across the UK. Our Test, Trace and Protect service is a collaborative service run by Public Health Wales, the health boards, the local authorities and Welsh Government. And we work together then in a networked approach to be able to follow up patients very quickly in Wales who have flagged positive. So the content of my presentation is I'm really going to concentrate only on the laboratory response and the expansion of testing and some of the challenges that we had with that. We do recognise the multidisciplinary approach to managing the pandemic and the various teams across healthcare and public health, not only in Public Health Wales but across the health boards as well, who have led and supported the response. So one of the first things that I really want to talk about is the timeline to molecular tests. So molecular testing being our primary service that we offer for this virus in the routine labs. And I just want to show you the difference between the timeline of the development of tests for SARS-CoV-1 versus SARS-CoV-2. So SARS-CoV-1 was first discovered in 2003, to be completely honest. This was after, in November 2002, we had a case of atypical pneumonia reported in Guangdong, China. Like I say, it wasn't until March the next year that they identified it as being associated with a coronavirus. And already by this time, we were seeing transmission in the community and global transmission of this virus. And it wasn't until April the 14th in 2003 that the genetic sequence was released by the US CDC. So you can see that actually there was quite a prolonged sort of a hold off essentially before we could actually get any genetic sequence to allow us to do any molecular testing. This is in stark contrast to what we're seeing now with SARS-CoV-2. So the virus was, we were first aware of it at the end of December 2019 when a report of pneumonia cases was reported in Wuhan, China. However, by January the 5th, 2020, we already knew that it was a new coronavirus which was distinct from the seasonal coronaviruses and indeed SARS-CoV-1. And in January the 12th, 2020, the genetic sequence was released by China itself. So we didn't have to wait for any sort of other external agency to do that work. China were very keen in supporting us with finding out what was causing their problems. And this means then that this opens the gateway for molecular testing to be developed. And one of the first ones was the Corman assay, which is widely used across Europe and globally. And they not only provided us with the blueprints, so the primers and the probes, based largely on work that had already been done successfully on MERS coronavirus by this same group. And so whilst we could use the genomic RNA as positive control, we had no positive control because the virus wasn't widely available. They could provide us with synthetic control, which could be used then to validate and verify the assays. An interesting note here is the order in which the tests were to be used. So we had a screening assay, which was the E-gene assay, which covered the sub-echo group of coronaviruses. So those are the bat species of the beta coronaviruses from which this virus emerged. And then you followed that with a confirmation assay, which is the RNA-dependent RNA polymerase gene. And then there was an additional confirmation assay should you require it. As you know, nowadays we rarely use single gene targets for this particular virus, opting either for a dual target or a triple target test. So implementing laboratory developed tests is normal for most emerging infections because commercial tests are simply not available, and these tests can generally be rapidly deployed. They offer quite extensive flexibility because you can adapt them for your local platforms and your own local workflow. So for example, if you wanted to use a different extraction platform than is described in the blueprint assay, then you can do that as long as you go through the validation process, and you can make it fit the workflow that you have in your own local laboratory. These first deployed assays are often the best characterized, and so you get to know how they work, you get to know all of their kinetics, and then you can use them then for the benchmark for other tests, and especially if these assays are used across other labs, because you can compare and contrast results using EQA and IQA. However, what we found certainly with this particular issue is that as we've relied more and more on commercial platforms to provide us with our molecular diagnostic capacity, it does mean that there is a significant skill shortage in development and implementing of these in-house tests. And this is certainly the case in Wales, where there's very few people now that are available to take these assays off of a paper and just run with them. And so this is something I think that retrospectively we should look at going forwards. This is one of the first assays that I run. So this is a serial dilution of RNA transcript run over time, and it showed really good sensitivity. I was able to get to the same level of sensitivity as was described in the Coleman paper, and as you can see here, there's very little non-specific reactivity at that lower edge. The kinetics was very good, which meant that I could put in an internal control very soon into the assay without affecting sensitivity and specificity too much. However, there was an announcement made by PHE that when they were rolling out their in-house testing, they would start using the RNA-dependent RNA polymerase assay. And as you recall from the previous slide, that is essentially the confirmation assay. So I was really confused about this because I really couldn't work out why they were doing this, because I had seen no problems. However, what they were reporting was a significant non-specific reactivity in the e-gene assay, and this was confirmed by other groups that I was in contact with at the time, where they were seeing low-level positivity at CTs of around 33, which were consistently reported across each of their runs. However, my concern was that I couldn't get the assay to perform as well as the e-gene assay in Cardiff, and so despite trying to re-optimize it to get it to work better, I still felt that the sensitivity wasn't good enough for us to use it as a frontline test, and so I used it, after re-optimization, I continued to use it as a confirmation assay in Wales. So this is the reasons I was so concerned. So you can see here in my test one, using the conditions that I had set up for the e-gene assay, you can see that the kinetics just doesn't seem to be as easily as consistent as for the e-gene assay. And indeed, what I was seeing was a little bit of noise towards the lower end, which can confuse staff when they're trying to interpret results, particularly when you've got large numbers going through. Our first positive in Wales was at the end of February, and this is what it looked like. So you can see here I've got the e-gene assay working really well, and I've got sort of a set of 3 controls there to the lower edge. So the yellow is at the lower end of detection of the assay, and you can see here in the RDRP gene assay that although it's detected really well, I have lost that lower end here, so the sensitivity just wasn't quite there for us. So I was quite happy to move forwards with this together with an internal control. What actually transpired was that there had been quite significant contamination of the primers and probes by the RNA transcript. So if you recall, there was a indication that you could buy transcript to help you verify those assays. And it turned out that what was happening was that people were requesting that the transcript be made by the same companies who were making the primary primers and probes. So contamination was getting into the system. So you were finding that manufacturers, when they were making assays, you were getting RNA transcript as well as the primer as well. So when you were running your tests, even if you run blank samples, you were still seeing positivity. And this was an ongoing problem with the in-house assays, because not only did it then affect the e-gene assay, it also affected some of the other target assays as well. Another thing that we needed to be mindful of as well is that mutations in primer and probe regions are quite extensive. So this is the original Coleman paper here showing the positions of where the primers and probes are. At the bottom 3, where you can see a lot of genetic diversity, these are the bat coronaviruses, and so you can see here very little in the e-gene, which makes them very good for detecting the Sarbeco group, and then you can see the discrimination across the other 2 assays. So it detects more the human strains rather than the bat strains. However, viral mutation does have quite a significant impact on testing. So we know and accept that viruses mutate over time, and the rates vary by nucleic acid type and the immune pressure. Coronaviruses do have some level of proofreading, and together with the production of subgenomic RNA, used to combine this proofreading and the subgenomic RNA, you protect that large 30K base genome, so you don't see so much variation as you might see with other RNA viruses. Most of those mutations that we see are synonymous, so that is that the downstream protein isn't affected, so they very rarely affect the virus in terms of its capability to infect, bearing in mind that we have seen spike protein changes which have affected infectivity certainly and recognition of the host receptor site. However, a single nucleotide change that might not affect a protein can have quite dramatic effect on your assay sensitivity, particularly if it happens at particular key areas in your primers. And so monitoring for these changes are challenging in a commercial system. However, at the beginning of the rollout of the testing and the commercial companies getting on board, if you read the small print in your IFUs that come with the kits, it might indicate to you which WHO assay was used as their blueprint. So there's a whole list of assays which have been used in development, and you can perhaps find out from those which ones are being used so that you can compare as the virus changes whether or not you're going to have issues. But as you know, as commercial companies come online, most of these tests become secret, so we're not able to get the sequences, so we still rely heavily on the commercial companies to keep an eye on that for us. We also then had the next pressure of the WHO announcing that we should test. And at this point, I think the pressure on the laboratories became extraordinary, and it caused a lot of issues in terms of staffing, in terms of throughput, in terms of, essentially making a service which would normally be doing routine testing for, a smaller number of people into almost a factory of COVID-19. We also struggled initially with some of the media and public scrutiny for the laboratories. There was a lot of certainly criticism raised at us that we weren't accelerating our testing fast enough, the numbers going through weren't fast enough, guidance was changing on a daily basis, so one moment we were only testing the community, then we were only testing the hospitals, and so we were trying to work alongside policy changes as well as increasing the capacity in the laboratory. We in Cardiff and across Wales, it's no secret we use dry swabs, and so to some extent we were less affected by some of the issues which would come further downstream, but we still had those problems. And interestingly, the media were very interested in the lab, so most of the stock photographs used around the UK to refer to laboratories were actually taken in the Cardiff laboratory. So all of these people here are in our lab in UHW. So I'm going to talk about the first real issue for us, and that's the global shortage of guanidine isothiocyanate. So in solution, that's also known as guanidinium thiocyanate. So this is a strong pleiotropic agent, and in solution, it's used for the isolation of nucleic acid. And it works in two parts. So the guanidinium cation and the thiocyanate anion work synergistically to break down proteins, and basically that includes clearing any nuclease activity. And what that allows us to do then is stabilize RNA in solution, so you can store RNA for long periods of time, and it allows that purification process of RNA in particular to be really robust and stable. And it fully denaturizes viruses because of that sort of that dual effect of the guanidinium and thiocyanate. You can denature viruses, and for us it works well with dry swabs. We essentially take a swab and the RNA is fixed to the swab, or the virus is fixed to the swab, and then we can break it into the guanidinium, and it is then released, and it really does store quite well. However, there was a global shortage. So one of the first things I did was so GITC is the other name that we would call it. It went into UK allocation, so all the commercial companies that were producing it ended up having to allocate their limited supplies, because as the world accelerated their testing, guanidinium being a key part in most lysis buffers became so short that we had to basically ration it out. However, this was our key ingredient for inactivation of the virus, bearing in mind that we were working with a category 3 pathogen, so we had to make sure that the downstream applications, the extractions, was performed safely. So what we did was, so this is the paper that the original recipe, so the L6 buffer is described in, the Boom paper, and I disseminated this to lots of universities and local pharmaceutical companies to support our supply chain. So we asked them to make it for us. However, we didn't always go to plan, because even though we were acceptance testing it as it was coming back in, we did see contamination as you move out of the sort of normal routine pharmaceutical companies that make these things. So, for example, one source contaminated a batch with Amplicon. They were making a local PCR, and somebody accidentally released Amplicon into the same room where they were making their guanidinium for us. Unfortunately, this only affected one of our platforms, so when we were doing the acceptance testing, we had to check it across all platforms going forwards, because we missed it initially, because the platform we were using for acceptance testing wasn't the same as the Amplicon. And then we had another source which was contaminated via an infected worker, and obviously we can pick that up really easily because it's the whole virus, and it would be positive in all of our platforms. And so that's just one of the issues that we had, but it really did impact us greatly. So our response in Wales to facilitate an exponential rise, so this is just the stepwise things that happened that we could do to get our network up and running. And initially, it was just our sexual health routine services that were the first to be dramatically affected. So this meant that we stopped testing for chlamydia and gonorrhea on a regular basis, and we were doing maybe 1 or 2 runs every fortnight. Because we needed to deploy all of our molecular platforms, switch them over to molecular testing for SARS-CoV-2. And as you can see here, so we started with C-Gene, we were doing a bit of work with that, so we were using that as our main extraction platform, as EasyMag, which is the main platform that we were using, was reduced. We weren't able to get the stocks for that. The Roche system came online, so like I say, the chlamydia testing stopped, so we could revert Roche to that. We introduced a whole new platform. PerkinElmer came in. The Abbott M2000, which is used for our bloodborne virus monitoring, was switched to COVID testing. So again, there's another impact on one of our routine services. We introduced Cepheid. We introduced BioFire, the Neri. So these are the black box technologies to get a rapid result out. We had this in-house assay which we were still using as our gold standard to compare everything against. We were still being affected by primer and probe quality issues, so every time we were having to acceptance test, we were rejecting a number of batches due to this problem, so it was really difficult to keep it going. So we made a decision to put Starlets, which are the C-Gene platforms, and Nimbuses into all of the other labs in Wales. This was massive expansion of a single assay, bearing in mind that most of the molecular testing done in the routine labs around Wales isn't to the same extent as it is in Cardiff, so the expertise wasn't there. So we had this huge expansion of molecular testing, but in some labs that had no expertise before. We had 2 labs built, so IP5 Lab 1 was built, but then was diverted to the DHSC to be used as their lighthouse lab. So we built IP5 Lab 2, and we equipped and set that up for high throughput testing. And now we manage 3 sites dedicated to testing SARS-CoV-2, and we have oversight of the validation work of the entire network. So if we talk about quality, we have to do a network approach because we are UCAS accredited as a network. So for process sample to answer, we made mock dry swabs using pooled control material, and we sent those out across the network to make sure that they got the same answer across the way. We also then made a single panel containing 10 samples with varying CT values down to 35 for testing across all of the platforms used in Wales to make sure that everything was comparable and working well. We used the C-Gene assay, we moved over from the in-house because of all the problems we were having to the C-Gene assay, as it was the only assay in Wales that was testing for all 3 genes at the time. Most of the others are dual or single gene targets. And as the system was implemented in any lab in Wales, this panel was provided for testing in duplicate. So then those results were collated by us centrally in Cardiff to check for any issues. And this was an enormous piece of work done by very small numbers of people. So these are the sort of samples that we saw. So this is sample 4 of that panel. We have C gene when we made the pool. We freeze thaw it to send it out, and we just show that there's no change there. But then you can sort of see that across the different platforms you get very similar results, very little sort of change, and the detection was comparable across all of the platforms that we were being used for this particular sample. QCMD we performed very well, so we run QCMD across all of the platforms once that was available, and again we got good results and good returns. And then the issues really were at the lower level of detection, where this is where we saw lots and lots of variations. So these are the C gene results at the start, so 35, 36, 33 for the 3 targets in Cardiff. Slight change in Bangor, but not significant. But then you can see as you go into the Cephids, the Aries, Luminex Next Tag, and the Roche systems, at that point you would see flexibility. And we also saw not detected results for things like Hologic and Perkinelmer at that low level, lots and lots of variation. So it's very difficult to pinpoint what the lower level of detection was for most of the assays. We also validated other sample types. We did sputum and lavage locally. We did whole blood from recovering patients who were donating plasma to ensure viral clearance. So that was a set of work which we were doing in parallel. We also validated paraffin embedded samples as well, so that we could go back and do some extraction work on that. We validated and implemented serology, and we also supported the validation of some of the lateral flow devices which were deployed across various health boards. So there was a lot of work ongoing and a lot of pressure. In all of this, we also had to protect the genomics funding as well. So we already started pre-COG in Wales. We had some funding for the first 100 cases by Public Health Wales, and we sequenced and uploaded the first case to GISAID about 2 days after detection. Our initial process of collecting 2 swabs per case meant that the stored dry swabs, so if you remember from the laboratory picture, there's lots of sweetie jars full of swabs, they could be used specifically for genomics. There was no need for this to use the same sample. However, going forwards, once that stopped, we do have this difficulty in maintaining the flow of samples, the extraction capability, and the integrity of those samples and extracts going forwards, because we're centralised in Cardiff for the whole genome sequencing, and so any delays can see RNA degradation, certainly from extract. So in a perfect world, this is what we would want. You'd want a pre-analytical, which is primarily the same for all options. You'd have open extraction systems that you could feed into any of your downstream amplification processes. You'd have your black box technology working. You'd have your Roches doing their capacity, and you'd have your M2000. So in an ideal world in wave 1, this is what we wanted to see, and it meant then that we could get over 2,000 results available in about an 8 to 10 hour shift if we were able to use the systems to their reasonable capacity. The reality was more this, however, with the global demand in guanidinium thiocyanate required for our lysis buffer, our pre-analytical steps were often challenged, meaning that our throughput was slow. We were unable to get supplies from some of the companies, so even our business as usual levels were never maintained. We couldn't get stock from certain companies, and then a lot of companies put it through to the UK allocation, and so we were allocated what was available on a weekly basis. So for example, if we wanted to do 4,000 tests per day, often the allocation was 4,000 tests per week. And so you can see that our limitations were quite grand. And interestingly, again, so even though I'd gone for a platform which perhaps wasn't used as widely in the UK, we were getting an erratic supply chain from South Korea due to the fact that flights weren't coming. They were suspending imports, certainly from the Far East. So we were generally challenged. So our potential throughput of over 2,000 tests today, in reality, in a good win behind us, was only around 950. The other thing that we had was staffing issues. So if we look at pre-pandemic, the clinical virology team, there's myself and Rachel, who are what we would call ticket holding virologists. And at that time, I was only half-time a consultant because I work predominantly in surveillance and development. We had two microbiologists who were working with us at the time with interest in virology, who were really supporting us, certainly in terms of the deployment of the assays, reporting, and also in the routine testing. We had a senior virology ID consultant, trainee registrar working with us, and in March 2020 we got an advanced nurse practitioner to help with us. And then the laboratory team itself, there was just one Band 8B, the operational manager is Jonathan, and then we had our normal staff things. So during the first wave, we basically had initial support from laboratory staff who were furloughed because their laboratories were shutting down. So that would include the universities, and also some of the other public health laboratories came in to support us. So we had people support from microbiology, we had support from other disciplines in pathology to keep the service going. In the second wave, we relied quite predominantly on students. So students who were off university were coming in to support us. And then as we came into the third wave over winter, we took the students and we had bank staff from all disciplines, and we moved to a 24/7 service to try and keep the samples going through a day. The additional time was quite phenomenal. So as well as regular inputs into IMTs for our local regional response, we also supported with development of tests for in other sectors such as university. We also advised on sampling and testing logistics, and also supported the Lighthouse Lab setups. We support for Welsh Government meetings at various levels, including and supporting in the UK. So it's an extremely stressful time in terms of the amount of time used for meetings in general, as well as supporting the lab development. So in summary, the emergence of SARS-CoV-2 put unusual pressure on the laboratory in its capability to provide high throughput testing. It's the highest level of scrutiny that the laboratory has ever faced. Some of the negative reports in social media and in the general press were actually very difficult for some of the laboratory staff to deal with. Whilst molecular tests remain the frontline tests in the routine lab for acute infection, we were really uncomfortable with the increased use of using them outside of the scope of the IFU, in asymptomatic screening, and certainly in low prevalence areas where you're testing everybody. You will have seen presentations earlier in the series where you're talking about sensitivity and specificity affected by prevalence. And so what we need really going forward is always to have this clear guidance on what their limitations are and what you should be using the test for. We had some unique challenges in that we are accredited as a network, and so validation and verification and certification of the things needed to happen as a network. So that was an extra pressure. And a lot of our support teams who work in that area, so quality leads, were working from home. So most of this was done over Teams meetings. The rapid expansion and turnover of staff with very little laboratory experience did require extra input in terms of training and proficiency. And and bearing in mind, despite all of this, we did manage to get through both waves with reasonably intact, with a good throughput, and very few dataxes, thankfully, very few issues in terms of quality. We did manage to keep that going. So just to acknowledge, my acknowledgements are generally just everybody who's put on a lab coat to support the COVID-19 response, everybody who sat through back-to-back meetings to support the response, and anybody who's looked after a patient with or without COVID-19, and that depended on, of course, what the test said that day.
Oh, for goodness sake. Oh, hello. Thank you, Catherine. That was great. What a roller coaster it's been. The cupboard rubbing bare, the staff having to sort of develop away from things. So there's been a couple of questions. The first one is, how can we preserve and develop skills to run in-house assays? it seems that with UCAS, the effect has been driving us towards commercial assays, and actually, there's still a really strong need for in-house assays.
I think that's something that certainly the clinical scientists and biomedical scientists really need to think about. It's They're quite right. UCAS does seem to be driving it away. So all of our in-house assays that we had set up years ago, which are far more flexible in terms of changes in viruses and changes in pathogens, have largely been moved over to commercial assays because the skills just aren't there. People aren't being trained when they come in to analyze assays in the same way. I think it has to be embedded into the training programs of new scientists coming into the field, and that's at biomedical scientist and clinical scientist level, and learn the interpretation. I mean, we're getting older, aren't we, Kate? We're not going to be around forever. Those of us who know how to do these tests, I think it's just making sure that we train the next generation coming in as well.
Thank you. Yeah, no, there's certainly a lot of many more commercial assays out there. Since the next question is, since coronaviruses don't have a capsid, the membrane is predominantly composed of lipid. Certain surfactants have been demonstrated to be more effective disrupting lipid membranes. Which of those do you think would be best of the surfactants for coronaviruses?
Well, I mean, it does have a nucleic capsid. It just wraps around the nucleic acid, but it's so the lipid is the main part, which makes the virus stable and functions and infectious. But I mean, any surfactant will break up a lipid membrane if it's strong enough. I mean, if we're talking about laboratory stuff that you could perhaps use to inactivate the virus beforehand, things like Tritinex have been used. I know that that's now banned in the EU, I think, and so we're having to look at alternatives for that. But any surfactant, anything that will actually break it up, we just have to make sure that if we're going to be using it in downstream assays, that it doesn't affect the downstream assay. We use guanidinium because we know that it has effect on far wider than just the lipid viruses. But yeah, I mean, you use whatever you have, but like I say, Tritinex is the one that most people are using. But ferry washing up liquid if you really need to.
And Val has put in the chat, she said, you've mentioned that you supported the Lighthouse Labs. What sort of support was that?
So initially, right at the beginning, we talked about sampling techniques as well, and how to get samples to the Lighthouse Labs. This was right at the beginning, before we even knew Lighthouse Labs were being set up. So I was involved in some of those discussions. But then sort of latterly, certainly with the Newport laboratory, we did support with some of their validation work and inactivation studies, just to make sure that because we use dry swabs in Wales, that the virus transport media could be used as well locally. So it was just a case of helping them with some of their validation work initially.
Very good. And Mark Wilkes has asked, dry swabs seem such a good idea. Why doesn't everybody else use them?
I don't know. I don't know. We've tried. We've been using them, as you know, for decades in Wales. The problem that we are facing now, in complete honesty, is that you don't have virus to isolate from a dry swab. You put the dry swab into your lysis buffer, and essentially it's just for nucleic acid. And actually, we're going to have to probably step back a little bit from that. I never intended not to use virus transport media at all in Wales. There always was a plan that we would keep some, certainly isolates available for culture. I don't know why. The data that I've done some validation work now with Colindale, we know that the kill is very good on dry swabs, particularly the cotton cheap wooden ones, which is probably why you haven't been able to isolate virus off them if you use those instead of your plastic flock swabs. But yeah, it's stable, it's easy, you don't get leakage, and it works really well. And we did whole genome sequencing from them, and they'd been stored for many days, and still able to successfully get the genome. So something to consider, maybe.
And a final thing, not an easy question to finish up with, but you'll love this, is why was there so much criticism of the Corman-Drostrom assay? And it seemed a little conspiracy.
Oh, yeah. So that was a really interesting debate which happened. So the Corman assay was developed, and it targets, as I mentioned, the sarbecovirus assay, so the E gene, and I think a lot of the criticism was around cycling for more than 40 cycles and non-specific reactivity after that time, which anybody who works with molecular type technology knows that if you cycle longer than necessary, you get breakdown of your primers and your, particularly your probe, and you get non-specific reactivity. There was a lot of criticism about the speed and the fact that it hadn't been validated properly. Now, bearing in mind that the assay was largely based on the SARS-CoV-2 assay, which has been very widely, validated, it was validated across Europe very quickly. Lots of people were using it. Certainly the first DCDC meeting I joined, it was the primary assay used across Europe. So we weren't talking about single laboratories validating it or just them and then issuing it. It was used across, pretty much across the whole of Europe, and everybody was having good results apart from the contamination when that happened.
Yeah, and yeah, I think I mean, the thing we found is obviously the assay that we use largely in England and Scotland was the one based on the RDRP, which had one mismatch or two mismatches from what was originally described. But ultimately, that's the same with any assay with an RNA virus, is that it can change over time. And in lots of ways, one still feels that that assay, which was slightly less sensitive than some of the others, is a much better marker for, what's active infection. And the struggles we have now with sort of old bits of DNA that are hanging around in our PCRs and trying to interpret that. But anyway, that's another thing. So any other questions from anybody? I'm slightly not seeing this question. If you would change one thing in this rollout, what would it be?
Oh, gosh, with the benefit of hindsight, possibly would have rolled out a lot quicker. Probably would have, got other labs involved very much sooner. Got everybody up to scale sooner. The issue that you had, I mean, I don't think when we come back to reflect on all this and whatever happens afterwards, I don't think the decisions we made at the time could really be criticised. I think they were right and proper for the events that were happening at the time. Everything now is looking backwards. So, we know that if you're going to have to test 7,500 tests a day, that the systems you were choosing at that point for what you thought were never going to work. So I certainly wouldn't put all my eggs in one basket in terms of platforms. If I was going for a commercial platform, I'd get as many in as possible. But I think, like I say, it's easy to look backwards and reflect and change things. But and perhaps we could take that into the next issue or pandemic should we come into it. But I have a horrible feeling it's all going to be forgotten again. I just think that we'll remember the good parts and not the bad parts.
So somebody has pointed out that the push to commercial assays isn't UCAS, although I often find that UCAS is much happier, but it's EU regs. But we'll let that one somewhere in between all of that. The truth lies. What about platforms with single genes and multiple genes? What's your feeling about that? Are you happy with an assay with a single gene target?
If if we can show over time that the virus doesn't change in certain regions. I mean, the E gene is actually quite stable when you look at it, you know, over all of the variants. So yeah, I think what we're seeing, that a lot of the dropouts tend to be in things like nuclear capsid. We're seeing the dropouts in spike, really unexpected. Um, you know, I think over time we may find that sweet spot in the virus, which is very specific for SARS-CoV-2 and also very stable, where you could perhaps just start looking at a single gene. I think the multi-gene actually adds a level of complexity in terms of interpretation, particularly when you are missing one, or you've got, you know, you're not detecting them as well across the board. It's just finding that sweet spot at the moment. I don't think we're quite there yet, and I don't think the confidence would be quite there to move to single gene. Saying that, the e-gene assay is still used in Cardiff for the genomics, and it's consistently positive for everything that we find, you know, for the viruses which are coming through. So it does look reasonably stable.
Yeah, no, we have some assays that have remained stable the whole way through. Just and then next question, and I'm probably not getting all of these in order. Any thoughts on CT values and viral load in relation to prognostic factors or infectivity?
Well, I mean,
Prognostic first.
prognostic. To be honest, we're seeing at the moment with some of the new the imported cases coming in, the viral loads are, if you were to register your CT value in relation to your viral loads, they're coming in with, you know, CT values of 11 in some of our assays, and they're really well, you know, they don't get severe infection at all. Whereas we've had people in ITU going for ECMO who have died, whose initial samples in the acute phase have had, you know, less CT values. I don't think it's as black and white as people say. I don't think you can be really sure that, you know, that you can base a prognosis on what the CT value is. I think there's a trend, but I don't think it's we're you know, I wouldn't be comfortable unless we have a purely quantitative assay where we've got you know, a real idea of what that result might mean in the patient.
Thank you. And then I think we'll do one more question, and I should first of all just also announce to the audience that you got an MBE for all your work, and well deserved. So you know, credit to all the work. Wales should be very proud, and I'm sure they are. Have you found it hard to comply with health and safety guidance of the virus, and full compliance with the IFU's information for use for the CE marking of commercial assays?
Yes, yeah. I mean, we certainly, the IFUs, if you read them, all say you should not use them in asymptomatic people. You should use them in symptomatic people, and you should look at the clinical history. When you have huge numbers of samples being tested, you can't individually look at all of those results and interpret them. That is, you know, we are using them in a way that we wouldn't normally in a diagnostic field. They are being treated more like a screening assay, and really you then want to have an algorithm to support that screening strategy, which is different to diagnostics. So I am a little bit, you know, about that. In terms of safety, we've done our best. We really have done our best. It's a category 3 pathogen. We've tried to manage it as a category 3 pathogen. I would like to see it move down to category 2 now that we've got a good vaccine. We're now in the situation where, according to the rules, that if you have a known positive patient coming through, you should treat that sample in category 3, even though you're managing lots of ones that you don't know that are positive in cat 2 facilities with extra safety. I think we need to be pragmatic. I think we need to start looking at how we manage these samples in the lab in the background of having a highly vaccinated population. And only when we've got a suspected VOC that is immunoscope that perhaps we want to look at, you know, continuing that use. That was a particular challenge for us, and I think in terms of all the laboratory responses, they probably had that issue that your category 3 lab was filled up with samples, which is a risk in itself.
I mean, do you really think, I mean, that this virus needs to be handled as a diagnostic in category 3?
No.
I mean, in our, I mean, I'm probably in our lab, we've used it as I'm sure many labs have, and we do all the work in the cabinets in Cat2. But given the amount of positive samples that have been going through this lab, and they've all been in viral transport means, they've not been coming in inactivated, and the number of people working, and we've not had an incident in any of that, it does make you think that the processes, that you're doing in that Cat2 environment are by just a live experiment have been robust.
I completely agree with you, and I'm surprised it hasn't already been derogated in the laboratory, because it does stifle us, and I mean, how many samples have, I mean, the UK processed at the moment at this enhanced Cat2 level, as you say, everything in a cabinet, staff in their PPE, it's how many incidents have we actually had, and I think it's time that we were a bit more pragmatic, particularly with the rollout of the vaccine.
I mean, one of the other things that it sort of cascades to is using the tube system in, say, the hospital to get the samples to you. They all have to come by hand because they're not allowed in the tube system because they're in enhanced. And then it means that they get left sitting on a bench somewhere because the person to come and pick them up hasn't come, or the system hasn't. Whereas if you just put them in the tube in a little container, they'd arrive immediately. But maybe that's just us. But
no, I think we're too flippant. I don't know, but I just think it's time that we were a bit more, realistic.
yeah, I mean, do you know if there is a review of the safety the laboratory's safety requirements? I think because everyone's coping, they're probably not feeling that there needs to be a review, but I think it would be hugely helpful.
I think it would. I think they've not seen what it's like at the coalface when you do have, samples that go missing. That is an issue. And getting the samples to us is an issue. We still have to bring couriers. We still have to, have, like I say, the rules are if it's known positive, you're supposed to handle it in a Category 3 lab. Stupid.
Very good. So I think there are some more questions, but I'm not sure I've completely got the master of this system yet. So there's somebody wondering if you're using dry swabs with antigen testing.
We've had a look at it. Yeah, we were having a look at it. I'm not sure how well the antigen will cope on the swab, to be honest with you, and I don't know how quickly we would see degradation of the virus, because you'd need to have it reasonably intact. It is something that we are looking at, though, because we've got an antigen test which we're looking at for one of our platforms. Just to have a look, really. But yeah, I'm not sure how reliable it'll be, but we'll look.
Very good. And Bristol PHE use special new pods. This is why I came to this meeting. That I can't see the next line, but I expect that they do something clever. Let me see if I can get this. Oh, that are sealed, so we can pod them now instead of in the old system.
Yeah, but wouldn't you just love to be able to use the old system and not have to buy new pods?
Well, this is true. Next question. This is a lovely question. Where do you see testing in 6 to 12 months' time?
Oh, I would really love this virus just to be part of our normal routine with business as usual. Will that happen in the next 6 to 12 months? I very much doubt it. We are not in a normal world, really, are we? Where decisions are made, rationally, I'd like to say. I think it'll take time, but eventually this virus is going to become business as usual for us. I would like to see it in 12 months' time. Whether it will be in 6 months' time for the next winter, I doubt it. I think we'll still be testing anybody who breathes in the street. I'd like to think we wouldn't be, but I suspect that's the reality, and I think we've got to prepare ourselves for that.
Yeah, I agree. I think we need to get to this time next year before the amount of testing will decrease. Who's going to stop testing care home workers every week until we've been through another winter? What policy document's going to say that? They're just not. So there's a lot of testing that's going to carry on. Do you think there could be more just use of lateral flow though?
Well, I think that's certainly the direction of travel. I mean, whether or not you know, the data will support that. Certainly, as we remain, if we stay in a low prevalence population, you know, and it will be more lateral flows. I mean, I'm slightly concerned about the plastic mountain that we're leaving with that. I am very sort of ecologically aware of the amount of plastics that we've used in this pandemic, just as we go into a world when we're trying to stop single-use plastics. But you know, that's just me on my environmental soapbox. But it's, yeah, I suspect that'll be the direction of travel. We'll be using lateral flow more often.
My all our electricity is wind powered here and an electric car, but yeah. But I have an old house, so all the heat just goes out the doors.
So maybe you could use some lateral flow cladding.
Yeah, right. I think, Catherine, you've done very well answering all my questions. I'm just double checking with all these little chats and things like that. Oh, here's another one. How have you used the inexperienced staff seconded from universities?
So
I don't
mostly
know whether
they,
they're always inexperienced, but
no,
that's
no, yeah, they generally had lab experience. So there was two sort of cohorts. So we had a cohort who were very sort of fairly inexperienced, but so they opened boxes and they helped with the numbering up and they helped with the matching of the samples, and you know, they did some of the what they called snapping of swabs in the lab, so snapping them into the lysis buffer. And then we also had them who had some experience. We had them running this machinery as well, getting the amplification done. It was just depended on what their skill set was as they came in, and not all of them stayed for long. Some of them stayed for longer. You know, the turnaround was quite incredible, and my hats off to all of the BMS staff who, you know, maintained all the proficiencies and trained all these people who as they were coming through the system.